So I'm going to start with this first video first I'll start with a little overview of protein synthesis, and then I'm going to talk about the first stage or the first step of protein synthesis, which is transcription. So I'm here in your notes where we left off. I'll go back and talk about RNA. I'm here at the top of this page. Protein synthesis happens in 3 steps. Now I will say, or 3 stages, I guess you could, no, 3 steps. One thing I do want to note is that those 3 steps are for eukaryotes only. Prokaryotes carry out transcription and translation, but do not do RNA modification. So let's just say that out loud in the beginning. Here's a nice graphic showing the overview of protein synthesis. In eukaryotic cells, it begins inside the nucleus. Here's our DNA, and the process of transcription is just making RNA from DNA. That's what we'll talk about first in this video. And then in the next video, I'm going to talk about RNA modification. That's eukaryotes only, the second step. And then in the third step, I'm going to talk about translation. That happens in the cytoplasm in both eukaryotes and prokaryotes. And it is the job of the ribosome. So we'll talk about that last. So let's start by talking about transcription. Now, in order to start talking about it, we need to get some terminology out of the way. So first of all, let's just define what transcription is. It's just the production of messenger RNA from DNA, and it occurs in 3 stages. So I'm going to talk about the processes within the overall process of protein synthesis. I'm going to talk about the steps of protein synthesis: transcription, first step; RNA modification, second step; and then translation, third step. But within those steps, I'm going to talk about the different parts of those processes as stages. So I'm going to use those different terms. Now, they're somewhat arbitrary. In any case, in order to talk about transcription, we need to get a little bit of terminology out of the way so we can talk about it. And I'm going to define 3 different things, 3 different components of step 1 of protein synthesis, which is the transcription unit, transcription factors, and the transcription initiation complex. So let's go here to this graphic. You have this graphic in your notes. You can label as I go along. So let's define 3 important components of transcription, the first step of protein synthesis. So first of all, the blue is DNA, and you can see here that it's in its double helix form. And the first important term is the term transcript. The transcript, or transcription unit, sorry, not transcript, the transcription unit is the section of DNA that's going to be transcribed into messenger RNA, which is the transcript. So the transcription unit is a DNA sequence, and that's just the gene. That's what's going to be transcribed into RNA. So that's the transcription unit. Transcription factors are the proteins required to start the process. And they're shown here in this light purple. These are called mediator proteins, but there's lots of different transcription factors, and they're really important for the beginning of what we call the initiation stage of transcription. And then like I said, they're shown here. Now, the last term that I want you to know, or you need to know, is the transcription initiation complex. So the transcription initiation complex is composed of these transcription factors. You can see them right here. And then you can see the DNA, and then these mediator proteins. The transcription initiation complex, the purpose of that complex is to allow for the binding of the enzyme that's responsible for building the RNA. And that's just RNA polymerase. So we had DNA polymerase that was building DNA. And now we're talking about, before we were just talking about making more DNA, now we're actually talking about the function of DNA. So what does DNA actually do? And DNA acts as the template for proteins. And so in protein synthesis, we're actually opening up and transcribing the genes so they can be translated into proteins. Okay, so excuse me, this transcription initiation complex is like a massive birthday party, and the process of transcription cannot begin unless everybody's at the birthday party. And that big complex of birthday party attendees is called the transcription initiation complex. Okay, so let's now go on to talk about the stages of transcription. And the first stage of transcription is called initiation. So we can talk about the transcription initiation complex here. So the first stage is obviously the RNA polymerase, which is the enzyme that's going to build the messenger RNA, needs to bind to the DNA. And the sequence of DNA, there's a binding site. Remember, it's an enzyme, so there's a very specific binding site. And the binding site for the RNA polymerase is on the DNA is called the promoter. Okay, a little bit more terminology. Let's add this to our notes right now. Here's, again, a stretch of DNA. Now, we haven't opened up the RNA. Nothing's happened yet. So we're just, again, defining the components, and we're going to try to start transcription. That's why this first stage is called initiation. And the purpose of this initiation stage is for RNA polymerase, we'll call it RNA pol, just like we called it DNA pol, but now the enzyme is making RNA, so we're going to change that modifier of the enzyme to reflect the fact that it's making a polymer of RNA. It's going to bind to the promoter, which is just all that the promoter is a DNA sequence where the RNA polymerase binds. Now, when I said I was going to introduce more terminology, we've talked about the fact that DNA polymerase is, that nucleotides have direction. And the direction of transcription is always going to be in this direction. This is showing you the direction of transcription. In that direction, we call, as RNA polymerase moves down the code, we call that going downstream. So you can think of it as a river almost. So the reason I'm mentioning it now is because the promoter is always just what we call upstream of the transcription unit. So here's our transcription unit, what we talked about up here, same thing. Now up here it's shown in blue, down here it's shown in green. And then we have this promoter sequence. So it's just A's, T's, C's, and G's where the RNA polymerase is going to bind. And that's what this is showing. This big red rectangle is RNA polymerase. Now there's also, just upstream of that, there's going to be a bunch of regulatory sequences. Some of those are actually going to be quite upstream. They're shown here. They're called enhancer regions, and they're actually all just DNA sequences. Remember, or not remember, but just to note, those enhancer regions, that's where those some of the transcription factors are going to bind. This whole entire double helix is going to come around and do this hairpin turn. There's specific proteins that are going to cause a specific arc of bending, and that's going to start the whole process. So that the whole transcription initiation complex is going to all be bound in here with the DNA kind of coming around and looping. You can go ahead and label the diagram here. Our transcription unit is also just, we call that the gene. And we're gonna try to make this messenger RNA. You can label this downstream. And then these are regulatory sites where we have these mediator proteins binding. Some of them bind to each other, but some of them bind to the sequence of DNA. And then upstream here, we have these enhancers, which are really just more regulatory sequences. In any case, the one interesting thing about the promoter is that there is a specific type of promoter sequence that's often found just upstream or within the promoter, only in eukaryotic cells, called the ta-ta box. So first of all, it's super fun to say. Let's say it a few times, ta-ta box. But the reason it's called the ta-ta box is because it's a bunch of T's and A's. T, T, A, T, A, A. Not that many. It's a relatively short sequence. But it's actually it tells the transcription initiation complex, and it tells RNA polymerase where the beginning of the transcription unit is. So it's really important. It's a very highly conserved. A quarter of human genes incorporate the ta-ta box into the promoter region. So it's really interesting. If we looked at our phylogenetic tree with our 3 domains, we've got the ta-ta box up here. Again, only eukaryotes, but a lot of eukaryotic genes, not just human genes, but a lot of eukaryotic genes have the ta-ta box. And like I said, it's a specific sequence within the promoter, in like a quarter of the promoter. So it's an important sequence. Anyway, so the first stage of transcription is to begin the process. So it's the whole thing, this whole entire birthday party. Again, remember how needy DNA polymerase was. Well, RNA polymerase needs a whole party before it'll even start the whole situation. So that's what initiation is. It's the formation of the transcription initiation complex, and then RNA polymerase will bind to the promoter. That's the first step, and we call that initiation. Now, the second step is called elongation, and that's just making of the messenger RNA. Now, RNA polymerase is needy because it needs the whole transcription initiation complex, but it's not as needy as DNA polymerase. RNA polymerase is going to unzip the DNA as it adds ribonucleotides, just using base pairing rules. So it doesn't need the helicase thing like DNA polymerase did. RNA polymerase is going to unzip as it adds. Now, it has some other differences as compared to DNA polymerase. First of all, using base pairing rules, across from every A, it's going to add a U instead of a T. So the A would be on the coding strand of DNA, and then on the messenger RNA that's snaking off, you can see it right here, it's going to be a U instead of a T because it's RNA, not DNA. And it's also not going to need a primer like DNA polymerase needed a primer. I mean, it's already RNA. Now, it's going to be the same or similar to DNA polymerase in that it always adds to the 3' end of the growing messenger RNA strand. And you can see that happening here. Here's the 3' end, and RNA, this isn't showing you RNA polymerase, but it would be right here. And then it would be adding, using, here's the template strand, it's showing you in red right here. And again, thinking about our the terminology upstream and downstream, RNA polymerase would be moving in this direction to transcribe the gene, or the transcription unit, right? This would be the transcription unit. And so we would call this downstream, and then therefore this would be upstream. Now, one thing to note is that, so this whole the process of transcription is going to be happening relatively quickly. Just downstream of the elongating messenger RNA, the DNA is going to the bases will rebind to each other. It's called re-anneal, and keeping the DNA as a single strand for really a very short time. So it's going to protect that code. So that's important to note. That the DNA will re-zip. It's called re-anneal. It doesn't stay open for very long. Okay, and then we get to stage 3. So how does the transcription unit know that it's done? How does the RNA detach? And that's what happens in the third stage, which we call termination. And in prokaryotic cells, contained in the quote-unquote end of the transcription unit, there'll be a termination sequence. So this RNA will be released when the termination sequence is reached. In eukaryotic cells, it's more complex than that. There's a signaling process and is outside the scope of this class. So you can simply write in your notes that termination is just when the messenger RNA is released. You could say from the DNA. And in prokaryotics, we have this prokaryotic cells, we have this prokaryotes, we have this terminator sequence. In eukaryotes, it's just more complex. You could just say that. Okay, that's it for the first step of protein synthesis, which we call transcription. And in eukaryotic cells, that's going to happen inside the nucleus. In prokaryotic cells, that'll happen at the site of the nucleoid in the cytoplasm. And I hope that was helpful. I'm going to talk in video 2 about RNA modification.